Hello, and praise God wherever you are. I want to believe that God is still taking good care of you. Um, Today we want to revise on paper 3, biology. That is a practical paper based on the West Kenya Union exam that we did last term, 2025. So before we do that, let's believe and pray. Heavenly Father, we come before you this particular time. Even as we want to revise paper 3 biology, be with us. Grant us knowledge and wisdom from above, spirit of understanding and even remembering. This is our prayer. In Jesus' name I pray. Amen. Okay, welcome to our revision. So learners, remember that paper 3 biology usually focuses on practical skills, and such kind of practicals includes food tests, specimen handling, interpretation of diagrams, and even ecological analysis. Remember, the goal here is usually to sharpen the observation, procedure understanding, and reasoning, which are the core of paper 3 exam. So always follow each step closely and think like a scientist during every setup. So to our first question of this paper, it was a hands-on question set from the food test, and remember you are usually provided with reagents, specimens, laboratory equipments. By the way, be well versed with the biological laboratory equipment. Know all of them. They are well taught in Form 1. So do a thorough revision on the equipment. Then also be keen with the reagents that you are provided with on your working desk, because those reagents will tell you or will even give you a clue on what you are supposed to do. So we have hydrogen peroxide there, specimen K, mortar and a pestle, 4 test tubes, scalpel, source of heat, and even test tube holder. So using a scalpel, we were to obtain 3 peeled cubes from specimen K, measuring about 1 cubic centimeters. For the first cube, we were required to boil it in water for 5 minutes. For the second cube, we were required to cut it into small pieces, then crush it into a paste. Then for the last cube, we were required to use it as it was, that is whole. The 3 test tubes were to be labeled as A, B, and C. Then to test tube A, we were to put the boiled cube, then add 2 milliliters of hydrogen peroxide and record our observation. To test tube B, we were to put the crushed paste and add 2 milliliters of hydrogen peroxide, then record our observations. To test tube C, we were to put the unboiled cube and add 2 milliliters of hydrogen peroxide, then record our observation. Now, remember specimen K was a potato, that is a plant. So one thing you should know is that this setup tests how catalase breaks down hydrogen peroxide to release oxygen gas. Remember, we are comparing 3 conditions. Boiled tissue, where enzymes are destroyed, crushed tissue, where we have maximum surface area, and then whole tissue. The form that is formed is oxygen. Remember the form, that kind of effervescence, that kind of bubbles that comes out, that is oxygen gas. This shows that there's some enzymatic activities taking place. Remember also, boiling denatures catalase, while crushing exposes more enzyme molecules to what? To the substrate. It demonstrates how enzymes are affected by temperature and even surface area. Now, what were the questions? So our first question here, we were to complete the table. That is an observation that will be taking place in each of the test tube. Now, in test tube A, you realize that there will be no effervescence, which you can also say no foam, or rather no bubbles. There's a reason for that. In test tube B, there were quite a lot of effervescence, a lot of bubbles, a lot of foam. Remember, this is the test tube that holds the specimen K that has been crushed. Then in test tube C, there is little or less effervescence. Now, let's account for the observation in test tube A. Remember, last time when you are doing paper 2, I told you there is usually a critical situation when you are accounting for something. You cannot account for something that you've not observed. So you must first of all make an observation, then account. As you are accounting, you are just giving reasons as to why the observation occurred that way, and then you justify your reason. Now, for example, here you see in test tube A, remember we are using boiled specimen. Boiling kills or denatures enzyme catalase, which we know enzyme catalase is the one that decomposes hydrogen peroxide. Hence, hydrogen peroxide is not broken down to oxygen and water. So learners, you have to understand that in each of the observations that we make, we differentiate the observation in each test tube. For example, we have no foam in test tube A, moderate foam in test tube C or less, and then we have highest foam in test tube B. So you see, this question kind of tests the ability to interpret results based on what? Experimental manipulation. Also put in mind that science is about comparing variables. Hence, you see temperature, crushing, and even enzymes presence. Those are some of the variables that are being compared in this question. Let's look at our next question. Between test tube B and C, in which test tube was the volume of foam produced the highest? Explain. Now, this question is 3 marks. So you have to first of all give the test tube in which there was highest foam, and then now give the reason as to why there was that what? Highest foam. So you realize that from the observations that we've made, test tube B is the one that contained highest foam. Reason: crushing increases the what? Crushing increases the surface area. Crushing increases the surface area for the action of enzyme catalase. You see, this will increase the decomposition of what? Hydrogen peroxide. Next question. Apart from temperature, state 2 other factors that affect the rate of enzyme-controlled reaction. There are quite a lot. Among them, we have pH, we have substrate concentration, enzyme concentration, coenzymes, and cofactors. Remember, pH, the acidity or alkalinity of something, affects enzymes, because different enzymes are suitable or work in different optimum pH, work best in different optimum pH. Then we have substrate concentration. The higher the substrate concentration, the higher the rate of what? Enzymatic activity. Same to enzyme concentration. When the enzyme concentration is high, we shall have higher rate of enzymatic activity. Then we have coenzymes and cofactors. Get to understand the difference between coenzyme and cofactors. Next question. Write a word equation for the reaction taking place in specimen K. Now again, remember this, we had such kind of a question in our paper 2. When we are talking of word equation, we need the words, not chemical. When we talk of chemical equation, we are requesting you to give the symbols, the chemical symbols. But here, since it's word equation, just give the word. So we have hydrogen peroxide here, which is being worked on by enzyme catalase. This is a reaction which will resulting to water plus what? Oxygen. So you write the words in full. You write the words in full. Next question. Name the process under investigation above and state where it occurs in human body. So the process there is detoxification. The process there is detoxification. As you can see, the process there is detoxification. And then where it occurs in the mammalian, it occurs in the liver, as it in all mammals. Remember, humans are also part of mammals. So when we talk of the place where detoxification takes place in human beings, it is usually in the liver. Okay, next question. What changes will be observed if an animal specimen was used in place of specimen K? Actually, there will be more effervescence, assuming that we used a piece of liver to carry out the same practical, then there will be more effervescence. There is a reason for that. So as you are giving an explanation for test tube A, remember that boiling destroys the structure of catalase, making it inactive. Without an active enzyme, hydrogen peroxide cannot decompose into water and oxygen. Therefore, this demonstrates that enzymes are protein in nature and sensitive to what? To heat. So when comparing B and C, remember that crushed tissue in B releases more foam because crushing increases the surface area for enzyme action. More enzyme molecules get exposed, speeding up hydrogen peroxide breakdown. This relates to the idea of reaction rate and exposed active sites. You see, the more the exposed active sites, the higher the rate of what? Reaction. Other factors affecting enzyme activity besides temperature, enzymes depends on pH, substrate concentration, enzyme concentration, coenzymes, and even cofactors. So get to know the difference between coenzymes and cofactor. Actually, learners, this question reinforces exam awareness because NEC usually love to test enzyme condition. In most of the practicals that you will go through in your revision, you realize that there is usually a test on enzyme condition. There is usually a test on enzyme condition. So when an animal is being used, an animal tissue is being used in place of plant tissue there, for this case we had a potato, assuming we are using liver, there will be more effervescence. Why? Because generally animal tissues have more catalase compared to what? To plant tissues, hence more higher rate of reaction or decomposition of hydrogen peroxide. So it will be higher in animal tissue compared to plant tissue. Okay, let's move to our next question. Question 2. Remember, in practical exam, usually have only 3 questions. So you've seen first question as is usually hands-on. When we talk of hands-on, we mean that this practical, you are doing it. You are involved in that practical. But you see now question 2 is a pictorial question, a pictorial question. The question states that study the photograph below and answer the questions that follow. So you can see this photograph here is actually it's just basically the photograph of heart. So this is just heart. And remember, heart is usually found in the circulatory system. But again, we have this one here. This one is actually what? This one is actually the lung. We have this lung and then we have this other lung here. So those are lungs. You see, lungs is usually found in the breathing system. The breathing system is also called respiratory system. So let's go to the questions and see what the questions state. State 2 organ system in which the 2 organs in the photograph above are found. Now, we have 2 organ systems represented here. But if you cannot identify the organs, you cannot identify the organ system. Remember the units of classification, all the way from the atoms, moving to the cell organelles, moving to the cells, then the cells combining to form tissues, then tissues combining to form organs, then organs combining to form organ systems. So you must know the organ systems found in the body of a living organism. Now, since here we have heart, you see we have heart here. This is heart. And then we have lungs. We also have blood vessels. We also have something like trachea. We have other organs and body parts. So the 2 organ systems involved here is number 1, we have circulatory system, and then number 2, we have respiratory system. Circulatory system is being catered for by the blood vessels and the heart, whereas respiratory system is being catered for by the what? The trachea and the lungs and all that. So always name the systems, not the organ. Name the system. You see, some majority of the learners were writing heart and lungs. That is not correct. Those are organs. Rather, they are not what? They are not the systems. So when you are asked for organ system, always give the systems, not the organs themselves. Okay. So next question. Label on the photograph the following structures: bronchi, left ventricle, septum, and then trachea. Now, I want us to be very keen with this diagram here, and more so, something that I want you to be very keen with is the bronchi. You see, bronchi is plural. Bronchi is plural. Therefore, when you are writing bronchi, when you are indicating bronchi, you have to indicate the two bronchs. The 2 bronchs. Because bronchs is 1. When there are 2, they are known as bronchi. So you have to indicate the 2. That's why you realize that these arrows, they come from the first bronchi here and then the second bronchi here, giving us bronchi. Okay. The next thing here is left ventricle. Of course, you can see the left ventricle is here, and it is usually the one that has more muscles. You'll just know it by the volume. So that is the left ventricle, and then we have the right ventricle. Then we have septum, as asked from the question. Septum is this muscle, this sheath of muscle that differentiates the 2 chambers of the heart. That is the right ventricle from the left ventricle. That is what is known as septum. Then we have trachea, representing the respiratory system. This is the trachea here. This one is the trachea here. This tube is the trachea. So always be accurate when you are labeling or when you are drawing. Be accurate. Then give the correct placement, because doing that shows that you understand the orientation and anatomy of living organism, which is basically the aim of biology, for you to understand the anatomy and orientation of living organism bodies. Okay. Next question. State one feature of the following structures. State one feature of the following structures and identification. You see the importance of the structure. All of the structures in the body of living organisms usually have the way that they are adapted to the function of that structure. For example, if we talk of maybe the teeth are structures, but they are usually adapted to perform their function of chewing or even biting or even cutting. You see, some of them have sharp edges, some of them have maybe they have ridges on the crown and all that. So in this case, we have left ventricle and trachea as our structures. So left ventricle usually have more thick and muscular wall, of course, as compared to the counterpart, which is the right ventricle. The importance, why does it have that thick and muscular wall? To generate pressure to pump blood, to distant body parts, or rather you can just say generates high pressure to pump blood for longer distance. Actually, left ventricle usually have thick muscles so that they can pump blood under higher pressure and then for longer distance. Number 2, here we have trachea. So we have trachea. Remember, trachea are usually C-shaped and they have cartilage, C-shaped cartilages, and then they also have cilia, and then they also have mucus secreting cells. So why do they have cartilage? Cartilage is there so as to prevent it from collapsing. Cilia helps in beating in waves, moving mucus to what? Pharynx. And then mucus secreting cells these are the cells that secretes mucus, which traps dust particles. Next question. Use an arrow to show the flow of carbon dioxide molecules through the chambers of the heart towards the lung. So this is it. You can see this is the arrow here. The arrow we are talking about, this is the arrow. Look at this arrow, the way it is moving from the right auricle to the right ventricle, then towards where? Towards where it is going to be refined, where carbon dioxide is going to be refined and released out of the body. Okay. So we move on to our next question. State one observable feature. State one observable features of lungs in the photograph above that suits them to their function. So from this diagram, you realize that we can see that we have so many alveoli, so many air-like air sacs. These ones, there are so many here, and you can see they are even folded. Where are they contained? They are actually contained in the lungs. So that is one of the adaptation of the lungs. one of the adaptation of the lungs. So we give our answer and say they have highly folded alveoli, which increases surface area for gaseous exchange, which increases surface area for gaseous exchange. Okay, so we move to our next question. Our next question, question 3, which happens to be our last question, comes from the topic ecology. Actually, you realize that most of the paper 3 biology that you'll be doing or that you've come across, always there is usually a question on ecology. Remember, ecology is basically the study of living organism in relation to the environment or surrounding. So you are provided with photograph A, B, C, D, and E. The organisms exist in a certain ecosystem. Remember an ecosystem, what's an ecosystem? Then study it and answer the questions that follow. So these are the photographs. Sometimes you can be given the real specimen to use in the lab. But here we have been given photographs. So you see, this is our first photograph A, then we have our second photograph B, then photograph C, then photograph D, then photograph E. Now, in as much as you can see this is a rat, don't use the name rat. And I've been telling students this one, that if you've not carried out a practical or a test to tell you that that is a rat, please don't say it is a rat. Just use the names given there. For example, this is specimen A. When you are giving your answer or when you are giving the food chain or even food web, just indicate it as specimen A, B, C, D, and even E. Don't use the actual names. From the specimen given and the photographs above, construct a food web of the organisms in the ecosystem. Remember, when we are talking of a food web, it must show all feeding relationships clearly. Food web is different from a food chain. Food web indicates all. How are these organisms relating in the ecosystem? Which one feeds on which one? Remember, how will you know an organism feeds on the other? For example, you look at this organism A. Is it an animal or a plant? You realize it's an animal. Which kind of an animal? Is it a primary consumer or a secondary consumer? And can it feed on organism B? Can it feed on organism C? What about D? What about E? So you realize that you take each organism and relate it to other organisms so that you can know whether it feeds on or it is being fed on. Now, take this note seriously. Take this note seriously that you must put into consideration that arrows must point to the eater. Arrows do point to the eater. And then you ensure that based on the marks given, you give at least basing on the marks given. For example, here we have 5 marks which are awarded. So we have to ensure that our food web has at least 5 relation. For example, A feeding on, you see we have organism A being fed on by E. So that one is one relationship, another relationship, another one, another one, another one, another one. So we have so many, but we need at least 5. So let's look at another question. From the food web, identify the longest food chain. So how do you identify the longest food chain? You just go back to the food web. You start from the producer. Our producer is D because it's a plant, so it is D. So you start from there. Then you see D. Which organism feeds on D? You see we have organism C. We have organism C feeding on D, organism C feeding on D, and then organism B feeding on D. Then from there, you now move to the primary consumers. C is being fed on by B, and then B is being fed on by E. So those are how many? D, C, B, E. Those are 4. The other option is D, B, E, which are only 3. So you realize that our longest food chain is basically D, C, B, and E. Okay, next question. Identify the organism with the highest biomass. When you talk of biomass, is the total dry weight. You realize that as the energy levels moves on or increases, there's a reduction in energy. Also, there's also a reduction in what? Biomass. So the organisms which usually have the highest biomass are usually the producers. Then next question, give a reason for your answer. Organism D occupies the least trophic level, as we've seen, and it is also the primary what? Producer, not consumer, producer. Now, other than feeding, how else does organism E benefit from organism D? So you first of all look at the organisms first. Organism D and then organism E. So you realize this is the organism D we are talking of, which is a plant and our primary producer. Then organism E is a consumer. But does it consume organism D? Probably not, but we might have other ways that they relate. So you realize that organism D produces oxygen used by organism E to survive. Because organism D is a plant, probably maize, it's a plant. And we all know that maize do carry out photosynthesis. So when they are carrying out photosynthesis, and then we have a gaseous exchange taking place, oxygen is being released as a what? As a byproduct. This oxygen is the one again being used by animals, such as organism E, which happens to be a cat, in their respiration. So you see, for survival, the organism E depends on D for the oxygen produced. Okay, let's move on to next question. The biomass of some above organisms is shown in the table below. We have organism B having a biomass of 250 grams per per square centimeter, and then organism C having 500, organism D 1000, and then organism E 100. So what is the question? Construct a pyramid of biomass to illustrate transfer of energy among the above organisms. Now, remember, as you draw the pyramid of biomass, the organism with the highest biomass forms the foundation, and then it increases as the pyramid moves up. You see now it will move basing on the organism with the highest biomass. So from D we shall move to C. Why? Because C has 500. Then from C we shall move to 250, which is organism B, and then organism E having the least with 100 grams per square centimeter. So the pyramid must visually represent energy lost at each level. Remember, why is this energy being lost? You see, for example, the primary producers, which have gotten this energy from the solar energy, that is from the sun, after converting the energy through photosynthesis into usable form, they have to use the energy to take care of their needs, the plants need, then they store some extra energy, and even we have another energy which is being lost to the environment in form of maybe radiation or even convection and so on. So the energy that will be passed on to the next consumer will be less compared to the energy that was absorbed from the sun. So that's why energy usually reduces as the levels increase. So this is typically the kind of graph you are supposed to have. For example, you see we have organism D forming the basis. Now there's a trick. How do we get the basis? You just draw, and then you get your origin from the midpoint. The origin or zero mark forms at the midpoint. Then you divide the biomass. For example, here we have 1,000. So you divide the biomass. So we have from the zero mark towards the right, we have 100, 200, 300, 400 moving towards that way. And then we have another half on the other side. So you see, for you to get 1,000, you take half of 1,000, which is 500, and then the other side also 500. Now you get a block of what? 1,000. The same thing will also happen to 500. You divide into half, you get it will be 250. So at this point we are having 250, and then at this point we are also having 250, giving us a block of 500. Same to 250, you divide by 2, and you also do the same to 100, which is for organism E. So again, after giving the organisms, always give the label the bars, label the bars representing the organisms. For example, the organism D represented by the first bar, organism C by the second bar, organism B by the third bar, then organism E by the fourth bar. So after doing that, you also ensure that you use a proper scale. For example, at this one, we are using a scale of 1 centimeter representing 100 grams per cubic centimeter. Okay, so up to that point, class, learners, we have reached to the end of our biology paper 3 revision. I want to congratulate all of you who have completed now paper 3 revision, and probably West Kenya Union exam in general, because we first began with paper 1, paper 2, and now we have done paper 3. If you've not revised paper 1 and 2, go to the channel, just click in paper 1 revision or paper 2 revision, and you'll get it, go through it, and give your reaction. If there's anywhere you've not understood, feel free to ask. If there's somewhere you have an additional, feel free to add your point. If you have a comment, you can also comment. Otherwise, continue to seek more revision materials and keep practicing. Remember, biology is a booster science, very, very interesting science, but it requires much of practice. Get so many revision materials and practice as much as possible. Otherwise, I want to wish you well, even as you continue revising biology. Goodbye.